moneris, which means single. And as the name suggests, the kingdom has in it all single-celled organisms. In the 1970s, genetic comparisons using small subunit ribosomal RNA highlighted a major problem with grouping all prokaryotic cells together under the single kingdom of Monera. So some prokaryotic cells, once considered bacteria, were actually more closely related to eukaryotes. So this required a larger phylogenetic category to be actually created. We called it the domain. So you have two domains, bacteria and archaea, and this is for the prokaryotic organisms, and then you have eukaryotic organisms which come under the domain eukarya. So what does this basically mean? Scientists realized that if they bunch in every prokaryote, into the first kingdom, the kingdom of Monera, it's not right, because bacteria and archaebacteria itself were vastly different. So what did they do to tackle this was that they created two domains. Okay, what were those two domains? Bacteria and archaea. Put these guys in two separate domains and then kept a third domain, called eukarya, where they put all the rest of the living organisms, which were all eukaryotes. So finally, what do you understand? There are three domains, two reserved for prokaryotes and one for eukaryotes. I hope it's clear. So the kingdom Monera consists of all prokaryotes, like we discussed, unicellular organisms lacking nuclear membranes. Prokaryotes are single-celled organisms with a cell wall, but they do not have membrane-bound organelles, right? So Monera can be further divided into two main groups. Archaebacteria, which are nothing but, the ancient bacteria able to live in extremely harsh conditions, and eubacteria, or true bacteria. Bacteria are the simplest, most primitive unicellular organisms. It's very small, with the length varying from 2 to 5 micrometers. So even if they are unicellular, they grow in colonies or filaments. Although it's just a single cell, they're amazingly complex and a fascinating group of creatures. And they have been found, in temperatures way above the boiling point that can make water evaporate in seconds. And even in the freezing cold, they eat everything from sugar and starch to sunlight, sulfur, and iron. In fact, there is this one species of bacteria called Deinococcus radiodurans that can withstand blasts of radiation 1,000 times greater than radium that could kill a human being. So each bacterial cell is made up of cell organelles with some nuclear material. So besides nuclear DNA in some bacteria, there is this extra little, chromosomal DNA that's present like a circle called a plasmid. Now, these plasmids are small, circular, double-stranded DNA molecules that separate from the genomic chromosome, and they replicate on their own. They replicate independently. So what exactly does this extra chromosomal DNA do in the bacterial cell? You might have heard that some bacteria develop resistance to certain antibiotics or develop toxicity towards them. Why does this happen? It's because of the same plasmids. So there are three types of plasmids which you find in bacterial cells. The first one is the F factor or fertility factor. And like the name suggests, it is for the transfer of genetic material from one bacterial cell to the other. The next factor is called the resistance factor or the R factor. So this is for, the bacteria to use for resistance against drugs. And finally, you have something called the cholecogenic factor, which produces colicins that kill other bacteria. Now, why would one kind of bacteria want to kill another kind of bacteria? What happens is that if you take the human body, for example, there are a lot of bacterial cells. In fact, around 90% of the cells of your body are bacterial. Now, before you freak out, let me tell you that most of these guys are good. They are there to help you out in various processes, for example, digestion. Okay, so what happens is if there is a bad bacteria which invades your body, the good bacteria need to go ahead and fight these, right? So they produce colicins, which actually go ahead and kill these bad bacteria. Another important question. Does a bacterium's cell wall, shape, way of moving, environment, really matter? It actually does. Because the more we know about bacteria, the more we're actually able to figure out how the good ones work for us or how we can actually go ahead and stop the dangerous ones from causing harm to us. And, for the most philosophical, scientific ones among us, bacteria actually help us answer important evolutionary questions and questions on the origin of Earth. So bacteria and their microbial cousins, the archaea, were the earliest forms of life on Earth, and they have played a crucial role in supporting, the larger life forms that we see today by developing photosynthesis and so on. In fact, you have cyanobacterial fossils dating back more than 3 billion years, and these photosynthetic bacteria have paved the way for today's algae and plants. So cyanobacteria, they grow in water where they produce so much of the oxygen that we actually go ahead and breathe. They were once considered a form of algae, and because of that, they also had a wrong name, blue-green algae, but they're not algae. Scientists believe that the more complex cells developed, as once free-living bacteria took up residence in other cells, eventually becoming the organelles in modern complex cells. Example, mitochondria. The cells which make energy for your body is an example of one such beautiful organelle, and you learn the history of the mitochondria in the chapter on cells. Let's now understand what bacteria eat. So bacteria can either make food on their own, that's when they're called autotrophs, or they can make food which is made by others, or they can eat others, and that's called heterotrophs. So autotrophic bacteria are again divided into two groups. One is photoautotrophs. Photo is for light. So what happens is they trap solar energy for utilizing carbon dioxide and use hydrogen sulfide in the place of water. So what happens over here? Oxygen is never released. The chemoautotrophs, chemo is for chemical, use chemicals by oxidation for food synthesis and carbon from carbon dioxide. The typical examples would be nitrifying bacteria, sulfur bacteria, and iron bacteria. Similarly, heterotrophs are also of two types, photoheterotrophs and chemoheterotrophs. So photoheterotrophs use light as an energy source and an organic compound as source of carbon. You might have heard of green and purple bacteria. They are a typical example. And then you have chemoheterotrophs, which use chemical as energy, and carbon may also be obtained from the same organic substances. So most of the bacteria actually belong to this group. Coming to respiration. Respiration in bacteria can be of two types. The two types that you know really well, aerobic, which requires oxygen, and anaerobic, which is in the absence of atmospheric oxygen. Let's now understand how bacteria, multiply, how they reproduce. As you know, they are prokaryotic organisms that reproduce asexually. So bacterial reproduction happens by a kind of cell division called binary fission. Now, binary fission results in the formation of two bacterial cells that are genetically identical. So what we're going to do now is we're going to visualize binary fission. So you have a cell over here, bacterial cell, and then there is a single DNA molecule which replicates, and both copies attach to the cell membrane like this. The cell membrane begins to grow between the two DNA molecules, and once the bacterium just about doubles in original size, the cell membrane starts to pinch inwards. A cell wall then starts forming between the two DNA molecules, dividing the original cell into two identical daughter cells. Binary fission is an effective way for bacteria to reproduce. But however, it's probably not the most effective way. Since the cells produced through this type of reproduction, are identical, the bacteria are susceptible to the same type of antibiotics. So to incorporate some sort of genetic variation, bacteria actually get a little manipulative and use a process called recombination. And bacterial recombination can be accomplished through conjugation, transformation, or transduction. So don't worry, they're all very easy. We're going to visualize it and do it one by one, and we'll do it in a way that you'll never forget what you've learned. So let's do the first one. It's called conjugation. So what happens is that some bacteria are capable of transferring pieces of their genes to other bacteria that they, come in contact with, kind of like give and take. During conjugation, one bacterium connects itself to another through a protein tube structure. We call that a pilus. Genes from one bacterium shift across to the other through this tube. Think of it as a handshake, where two bacteria meet up and genes get transferred from one side to the other. Next is transformation. So some bacteria are capable of taking up DNA from the environment. So these DNA remnants come commonly from, dead bacterial cells. Think of a sweeping happening, across the floor. A bacteria actually binds the DNA from all these dead cells and transports it across the cell membrane. The new DNA is then incorporated into the bacterial cell's DNA. So you understand the point here, right? We're getting genetic variation. Now, why is genetic variation important? Is because the bacteria's aim is to not be susceptible to antibiotics that we make for it. So if there is more variation, it will be tougher to create a specific antibiotic that would kill all the bacteria. We've all heard of a video or an image going viral, right? So where do you think that name came from or that metaphor came from? Nature, of course. So viruses are notorious of their ability to go ahead and hijack a host, invade it, hijack its cellular machinery and force it to actually build millions of copies of itself, that is of the virus. Now these copies are then released and then they go on to attack new hosts, spreading through populations. So viruses that infect bacteria are called bacteriophages. Now bacteriophage means bacteria eater. So sometimes instead of just infecting and hijacking the host, the virus picks up and transfers some of the host DNA. And this process is what we call, transduction. It's like making the most of your enemy, right? So transduction just cannot be this simple. So you'll have to have two types of transduction, generalized and specialized. So bacteriophages go about their work in a very gruesome way. The bacteriophage interaction with the bacteria involves a virus invasion, a hijack of the host to create copies of itself, and then a killing of the host cell by popping it open like a balloon. This is called lysis. So these new baby bacteriophages go on now to infect and kill other host cells. During the process, the bacteriophage cuts up the host chromosome into many small baby fragments. And when the baby bacteriophages are being assembled, some of these host DNA molecules, accidentally get packaged into the new viral particles, carried on to the new host cell where they are injected, and this actually causes genetic recombination. So this is the first type of transduction. Coming to the second type of transduction called specialized transduction, it's even more gruesome. So instead of just hijacking and killing the cell, the viral DNA goes dormant by incorporating itself into the bacterial DNA chromosome. So the cell continues to grow and divide, as if nothing happened. The incorporated viral DNA gets transferred to the clone offspring cells, and suddenly, in a nightmarish way, the bacteriophage reactivates itself, cuts itself free from the host genome, and hijacks the cell to produce new bacteriophages, which then lyse or kill the host cell and go on to infect additional cells. Let's now talk about the archaea. Archaea comes from the Greek word meaning ancient. And quite like its name, many archaea thrive in conditions that mimic that what was found more than 3.5 billion years ago on Earth. In fact, it wouldn't really fit into your or my definition of life as we call it today. They're more alien-like. But you know what? These guys have a good holding on our history. Back then, Earth was still covered by oceans that regularly leads to boiling point. Archaea thrive in the very same conditions even today. They're found in the most extreme of climates. Volcanic eruptions, hydrothermal vents, frigid Antarctic ice, super salty waters of the Dead Sea, and acid-laden streams around old mines. This is where you'll go ahead and find archaea. Some scientists call archaea living fossils because they provide hints about what the earliest forms of life, on Earth was like and how life actually evolved on our planet. What are the types of archaea? These simple guys also have subtypes and they're awesome. So I can't leave this discussion without talking about them because the habitats that they want and they thrive in are just so extreme. You'll understand how they got their names in just a bit. First up, you have thermophiles, which like unusually hot temperatures. So few species have actually been found to survive even beyond 110 degrees Celsius, and water boils at 100 degrees Celsius. Then you have psychrophiles, which like extremely cold temperatures, even down to minus 10 degrees. Then you have halophiles, which live in very, very salty habitats. So some can thrive in water that's 9% salt. Okay, and seawater contains only 0.9% salt for a comparison. Acidophiles, acidic conditions, and alkaliphiles, like the name goes, prefer alkaline environments. Archaea that populate extreme environments along with some of their bacterial cousins have developed some clever tricks and tools to actually go ahead and do so. For example, they produce special enzymes that help keep all parts of their cells intact, even in conditions that could make our human skin fall apart. So, because many archaeans thrive in these conditions, they're also called extremophiles, meaning creatures that love extreme conditions. So, the big question here is, how do they actually go ahead and do this? They make a variety of protective molecules and enzymes. For example, some archaeans live in highly acidic environments. If the acid got into the cell, it would destroy their DNA. So what do? They need to keep it out. So they have special molecules on their cellular surfaces that, come in contact with the acid, but are uniquely designed that it doesn't break apart. Archaeans that live in very salty water, similarly, are able to keep all the fluid from dissolving out of their cells by producing or pulling it from the outside solutes. Say like potassium chloride that balance the inside of the cell with the salty water outside. Now, not all archaea are extremophiles. Many live in more ordinary temperatures and conditions. For example, scientists can find archaeans alongside bacteria and algae floating about in the open ocean. So, domain bacteria includes the vast majority of everyday prokaryotes, the ones that you see and interact with on a daily basis. This can include pathogenic strains that cause diseases and beneficial ones which populate our foods and guts. So domain archaea, on the other hand, includes those prokaryotes that prefer to live in extreme environments reminiscent of early Earth conditions. Next up, we'll talk about cyanobacteria. These are also called blue-green algae. They are gram-positive prokaryotes. So they're photosynthetic and they have pigments like chlorophyll, carotenoids, and some awesome-sounding phycobilins. They have thrived in, like every environment, hot springs and moist soils, just to take an example. These guys have stood the test of time. Their cells are actually larger than normal bacterial cells. Their cell wall is multi-layered. It includes something called peptidoglycan, and they're photosynthetic pigments in the outer part of the protoplast. So some of these pigments lie freely in the cytoplasm. There are some minute granules which are attached to the photosynthetic pigment. These are called phycobilisomes. It contains the very same fancy-sounding photosynthetic pigment called phycobil. And now, let me introduce you to the smallest known organisms ever. They are facultative anaerobes. They completely lack a cell wall. But before you, start wondering, a facultative anaerobe is an organism that makes ATP by aerobic respiration if oxygen is present, but it's actually capable of switching to anaerobic respiration when, oxygen is absent. So, pretty cool, right? Mycoplasmas possess a plasma membrane. The smallest cell so far discovered is of Mycoplasmic gallispecticum, having a diameter of 0.1 microns. And, they're actually resistant to antibiotics that interfere with the integrity of the cell wall. Penicillins, cephalosporins, vancomycin, etc. And since mycoplasmas do not have a cell wall, they do not have a separate shape, they go against most of the typical rules that microbes fall in, and hence they're also called the jokers of microbiology. They are highly pathogenic and cause diseases like pneumonia, arthritis, and rheumatism in humans, and may stunt mulberry dwarf and bunchy top of papaya in plants. And to finish off, here's something interesting about mycoplasma. Much of the confusion early on in the research of bacteria was all thanks to Mycoplasma bovis and other related species because they were so small, just 0.2 to 0.8 micrometers in size. Because of this, mycoplasmas are now classified as the smallest self-replicating bacteria on Earth.